Another area where RECNAC research has been at the forefront is natural product therapeutics. As a result of this research thrust, RECNAC has developed and tested a non-toxic extract from a locally grown plant that is capable of halting new blood vessel growth and inhibiting tumor growth. RECNAC research has also developed a method for producing an immune stimulant from bacterial cultures that exhibits significantly anti-tumor activity. Dr. Xiaolong Meng, who came to us from China in 1991, has served not only as a senior RECNAC research scientist, but also as primary liaison with researchers in Beijing, where we carry out significant studies. Dr. Meng will tell us a bit more about the development of natural product therapeutics.
Okay, I'm going to talk about the natural organism extracts on cancer research. The natural organisms include the plants, bacteria, invertebrates. The purpose of this study is to find the high effective but low toxic anti-cancer agents. Here is how we did the study. The natural organisms were soaked in the buffers or solvents, the supernatant, are precipitated, centrifuged, filtrated, dried to create the crude extract. Then we analyze the crude extract for the components, do the cell culture for the cytotoxicity, do the X test for the angiogenesis inhibition, do the animal test for the tumor inhibition. Some of the crude extracts showed very good results for on these experiments. Then we separated them by their polarity, their size, their charge, to get the pure molecules. And then we repeat these experiments again until we find the high effective, low toxic anti-cancer agent. This slide shows one of our chromatography systems. Some of the crude extracts were analyzed and purified on this system. These are the buffer bottles, the pumps to drive the buffers and the samples through a column here, and then the samples can be purified and separated in the columns and detected by the UV detector, and the chart can be recorded in the recorder. Here is the collector to collect the peaks. We can separate the peaks for the further study. The chart from the recorder is called a chromatogram. The next slide shows the chromatogram from a sizing column for a plant extract. The extract passed through this sizing column. Then the big molecules came out first, and the small molecules came out later. They are separated. The separated peaks can be collected for the further study. This is just one example how we analyze and separate the extracts. This slide shows the differential cytotoxicity of an invertebrate extract. The y-axis is the live cells to control. The first column from the left is the normal cell line for the normal cell line. The second is for colon cancer cell line. The third one is melanoma cell line. The fourth one is the fibrosarcoma cell line. Any drug will kill both normal and the tumor cell lines at a certain concentration. The purpose of this study is to find an agent that kills more tumor cells but less normal cells. After treated with our invertebrate extract for 7 days, only 3% of the tumor cells were alive, while the alive normal cells were 45%. So the alive normal cells were 15 times than live tumor cells. This extract shows high selective cytotoxicity to the tumor cells. Another way to find an anti-cancer agent is angiogenesis inhibition study. Angiogenesis is a kind of blood vessel starting and growing. It happens around all malignant tumors. If any drug can stop the angiogenesis around the tumor, It may stop the growth and the metastasis of the tumor. This picture shows the fertilized eggs we use for the angiogenesis study. We add the tumor cells on the membrane and place the tested agents around the tumor cells. The tumor cells can stimulate the blood vessel growth in the membrane. If any tested agents can stop this growth, it is the angiogenesis inhibitor. This slide shows the image for the angiogenesis study. The top picture shows the angiogenesis. Many blood vessels grow around the tumor. The lower picture shows the angiogenesis inhibitor by a plant extract at lower concentration. And this right picture shows the incubation at a higher concentration. Not too many blood vessels around the tumor site compared to this one. This slide shows the angiogenesis incubation by a plant extract. At 50 microgram, the angiogenesis was 18%. At 100 level, it's 55%. 200 level, 73%. So this plant extract showed a very good angiogenesis inhibition and dose response. The hopeful extracts were tested on animals finally. We have done two kinds of animal studies. One is for the tumor incubation. The tumor cells were inoculated on the mice first. And then for the control group, normal saline were injected. For the treating group, we injected the tested agents in normal saline. 2 weeks later, the tumors were rejected and calculate the tumor inhibition. The other tumor animal test is the LD50, 50% lethal dosage test. This test for the toxicity of the agents. The higher the dosage, the lower the toxicity. The range of the low toxicity is from 500 to 5,000 milligram per kilogram. This slide shows the tumor incubation of a plant extracts on 3 animal models. The y-axis shows the tumor weight. The green color shows is for the control groups. The red color is for the treating groups. For the sarcoma model, the tumor incubation was 77%. For the lung cancer model, the incubation was 62%. For the melanoma model, the tumor incubation was 53%. This slide shows the tumor incubation of an invertebrate extracts on 2 animal models. For the sarcoma model, the incubation was 50%. For the melanoma model, the incubation was 43%. This slide shows the incubation of a bacteria extract on the sarcoma mouse model. The incubation was 80%. Those tumors are growing very fast on these animals. After the tumor cells inoculation, tumors will grow to 10% of the body weight in just 2 weeks. If a tested agents can reach 30 to 40% of the tumor inhibition on these models, it can be considered a hopeful anti-cancer agents in clinic. Some of our agents reached more than 80%. So they are very hopeful anti-cancer agent. We have got high tumor inhibition. We also want low toxicity to animals. This chart shows the LD50 test. The higher the LD50, the lower the toxicity. After giving the bacteria extract to about 4,200 milligram per kilogram, 50% mice died. For the comparison, the cyclophosphamide makes 50% mice dead at only 160 milligram per kilogram. So 4,200, 100. So the toxicity of our bacteria extract was very low. The daily dosage of this bacteria extract for 80% inhibition, tumor inhibition, was 12.5 milligram per kilogram. That's 0.4% to the LD50, while the cyclophosphamide, the daily dose for the cyclophosphamide is 2% to LD50. The cyclophosphamide is a chemotherapeutic agent used in the clinics. So this bacteria extract had more safety room than the cyclophosphamide. In summary, our extracts from the plants, bacteria, and the invertebrates shows a very high inhibition to tumors and a low toxicity to normal tissue and animals. They are hopeful anti-cancer agents in future. Thank you.